Hello, and welcome to this Nature Research custom media webcast titled How In Vitro 3D Models Are Influencing Infectious Disease Research. My name is Nikki Forrester, and I will be your moderator. Today's webcast is produced by Charles River. The inability of 2D cell culture systems to fully translate preclinical drug behavior into meaningful clinical efficacy or safety is a key contributor to high attrition rates in drug discovery programs. As in vitro biological testing is one of the most crucial early steps in the development of novel therapeutics, advancement within this area plays a key role in the evolution of drug discovery. The development of 3D models has expanded over the last decade due to their potential to bridge the gap between effective preclinical drug screening and predicting therapeutic success in patient clinical trials. Now, the use of 3D organoids at the early stages of drug discovery processes can promote the selection of more efficacious and safer drug candidates. Today's webcast will present how organoid platforms can be used to identify novel antivirals, as well as to interrogate vaccine responses in complex human tissue models. We'll begin the webcast with a presentation from Dr. Ludovico Butti of Charles River. Next, we'll hear from Dr. Dan Roca of Charles River. We'll end the webcast with a Q&A. To ask a question, just type it in where it says Ask a Question, and then press Submit at any point during the webcast. We'll do our best to answer all of your questions today. And now, over to Dr. Butti.
Okay, thank you for the nice introduction. And in the next 45 minutes, Dan and I would like to discuss the use of 3D models in drug discovery. And we will focus in particular on the area of infection disease. So we'll start off with 3D models and how these can be positioned in drug discovery programs. And we will give particular attention to the cell source. That is one of the key components to build complex in vitro models. So in this regard, we will discuss the use of organoids, and in particular gastrointestinal organoids, to study the host pathogen interactions. And I'll give you a few examples of how this tool can be really powerful to study antimicrobials. In the second part of this seminar, we will switch gear and then we'll discuss how to build an in vitro model for adaptive immunity, as we believe that this is in general the immune system as a whole is a still missing element of the in vitro 3D model. So, given my interest in the gastrointestinal tract, let's first take a look at the intestine. And here in this electron scan microscopy image, you can see the villi that protrude in the lumen of the intestine. And then at the bottom, we have this small imagination, also called crypts. The work of others, including Hans Clever, has shown that at the bottom of the crypts is where the adult stem cell resides. These cells, support from Punnett cells, basically divide, and as they access the villi, differentiate in all these different epithelial lineage. Now, this is just to give an overview of the complexity of the structure of the intestine and also the different lineage components that are part of the epithelial layer of the intestine. And this can be hardly recapitulated by using cell lines that are cultured in a petri dish. So not too long ago, the lab of Hans Clever has shown that if you take the adult stem cell and you put them in culture using the right growth factors, those cells start to proliferate and differentiate. And this is what are now called organoids. And these organoids are important not only because they recapitulate many of the epithelial lineage that you see in vivo, but also because they acquire a 3D structure that is reminiscent of the intestine. For instance, the cells that are actively dividing, shown here by Ki67 staining, are located in a crypt-like structure similar to the in vivo condition, and then the cells that are instead more differentiate are facing the lumen of the organoids, very similar to the villi in the small intestine. So now the question is, how do we position organoids, or more in general 3D model, in drug discovery programs, and why do we need these advanced in vitro models? So here you're looking at a subway map that illustrate what it takes from early discovery, of when you have an idea in mind, and to bring this all the way to the preclinical phase. So it's quite a long journey, but at the same time, there are several opportunities along the way where 3D models can help to select a successful preclinical candidates. So for simplicity, let's just take the green light that brings directly to the clinical candidates. And historically, the journey starts with screening many compounds in a model that is quite simple and less perhaps physiological than the human. And then as you move along the way, the throughput or the number of compounds decrease, and the physiological relevance of the experimental model increase. And there is a tipping point around here where we switch from the in vitro work to the animal testing. So what we have learned from clinical trial, that this type of approach has a very high attrition rate. Often drug that goes to patients fail in clinical trial because either they are not efficacious or they are not safe for the patients. And in the second part of this talk, then we'll also illustrate how this percentage can look even more worrisome when it comes to vaccine development. This is also due to the lack of in vitro models that basically, in general, we don't have a good in vitro system that can predict the response in patients, in particular when it comes to the immune system. So this is what we would like to highlight in this seminar. If we bring 3D model with multilineage culture and include also the immune component already early on during the in vitro testing, this is where we can elevate the physiological relevance of the system that we are using. And then at the same time, we can help to de-risk compound that otherwise might fail when they arrive at a later stage of the programs. So, and here I just give an example where to position 3D model along the drug discovery programs. So again, bringing organoids, organ on a chip, and start to test the immune system at the early stage will help to select better compounds that will be more efficacious and also more safe. Now the question is, how do we know if organoids, organ on a chip, and all these other advanced models can actually be better predictive of patients' response? Short answer to this is that we don't have a full picture just yet. However, data that we have been collected so far look really, really promising. And to find an answer to this question, we need to take what we have learned from previous clinical trials and bring them back to the lab. So with the knowledge that we have already acquired, we can interrogate this system and understand what is their advantage of using this early on in drug discovery. Obviously, the FDA Modernization Act that the US government has passed in the late 2022, early 2023, has helped a lot to give an incentive to these initiatives. And here on the left, you're seeing the different efforts that at Charles River in early discovery and discovery we are taking in this direction. So as I mentioned already, at the beginning, we will, in this seminar, we will mainly focus on organoids from the gastrointestinal tract, and then on tonsil organoids to study the adaptive immune response in vitro. Obviously, these advanced models are not only important to generate more efficacious and more safe drugs, but also are important in the aim to reduce the number of animals in research. And I'm sure if you're following this seminar, you are aware of the 3R replacement, reduction, and refinement of animal work in research. And probably you are also an active member of these activities. At CRL, we also add an additional R that stands for responsibility, which is the Charles River 4R mission, because we truly believe that we are responsible to push this initiative forward to basically reduce the number of animals in research. Now, in this review, we basically describe the key components that are needed to build a 3D model. And it goes without saying that the cell source is come first. Obviously, the cell has to be reliable cells and also scalable. And then you need to put the cells in the right extracellular environment, like ECM, and also all the other components that are basically make the cell feel as in vivo. And also add the engineering part where through bioprinting, bioreactor, and also all the microfluidic system, basically you can recapitulate the physiology that you have in vivo. So as I said, we will give particular attention to the cell source, and I will now describe some of the work done using organoids in the infection disease area. So this is just an example of the heterogeneity of the organoids, and here we are looking at stomach organoids. Here on the left, just to remind you the structure of the stomach gland with all the different epithelial lineage that derives from the adult stem cell. And then here and at the bottom, you can appreciate that within the stomach organoids, we have different lineage marker within the same organoid. When it comes to validation, here is an example. This is a work that we published a few years back when we were looking at the activity of a tumor suppressor, ASP2, that is important to regulate the activity of P53 as well as to maintain the cell polarity in the epithelial cells. So here you can see the staining for ASP2 in green of a healthy donor, and this is the tissue, the stomach gland. And then from this donor, we derive organoids. And what you can appreciate is that the organoids have a very similar localization of ASP2, here shown in green, at the tight junction, very similar to what we have observed in vivo. Now, organoids have been used, there is a quite large literature of using organoids to study infection disease. A lot of work has been put to basically use organoids to cultivate pathogens that otherwise were not possible to grow in vitro with using cell line, to study the replication of the bacteria or viruses, tropism, study lineage tracing, and obviously also the interplay between innate and adaptive immunity with the epithelial components. In the next few slides, I will focus our attention on what we have learned in the drug discovery program to study the host pathogen interaction. So here you're looking at cells, a cell line that have been infected with a bacterium that is H. pylori. And here to the right, you see the same bacterium infecting a gastric organoid. So H. pylori, there is a bacterial pathogen that colonized the human stomach and is considered the first risk factor for stomach cancer. And you can appreciate when we infect a gastric cell line with this bacterium, virtually all the cells are infected by H. pylori, here staining green. On the other hand, if you look at the organoid infected with the same strain, now you start to appreciate that the bacterium localized in specific region of the organoids, suggesting that there is a specific tropism for the bacterium for certain type of cells. And the work from others group have shown that these bacterium can target the adult stem cell as well as the pit cells in the stomach. So in this work, we were looking at the activity of ASP2 during the infection with Helicobacter. And here you can see that upon infection, ASP2 relocalizes at the site where the bacterium is attached to the cell, while in non-infected cells or in non-infected organoids, the ASP2 nicely localized at the tight junction. And this is perhaps even more clear in this 3D reconstruction. Now, based on this observation, we decided to set up a screen to find small molecules that could inhibit this phenotype. And I should also mention, this is the first step that then will lead the cell to lose the cell polarity and acquire an epithelial to mesenchymal transition phenotype. So I won't go through the whole screening that we have done, but I will just mention that throughout this effort, we identified an enrichment for compounds that can inhibit EGFR or AKT, and when those are inhibited, basically we block this phenotype. So, and here it comes to the validation of the screening. And to do this, we did what is normally done in the field, a colony-informing assay, that is basically to assess the viability of the bacterium that infect the host, and basically you lyse the cell and the bacterium and plate in serial dilution the bacterium to see the colony that is still able to form. Now, this experiment was initially done using the gastric cell line, and what you can see here, it's quite disappointing result, because when we infect the cell with H. pylori and treat them with EGFR inhibitor, we didn't see any difference whatsoever. So despite the disappointing result, we went on and basically repeat the same experiment, this time using organoids. And I should just remind you that in this condition, the bacterium is in the lumen of the organoid, and to get nutrients and all what it needs to survive is forced to interact with the host. Now, if you look at the right part of these slides, are the result of the colony-forming assay in the organoids. And what you can see now is when we infect the cell with H. pylori, we retrieve viable bacteria. But now when we add EGFR inhibitor, now the number of viable bacteria that are retrieved statistically decrease. And this is true for EGFR as well when we use an AKT inhibitor. Interesting, if we target a downstream pathway of EGFR, in this case ERK, we don't see this effect anymore. So, but this is just an example of when we use a more complex model, this can help us to identify compounds, meaningful compounds, that otherwise will have been considered as a false positive. Based on this result, or this result actually gave us the idea to extend this and use organoids as a platform for antivirals. And in this work, we use, instead of using adult stem cell derived organoids, we differentiate them from iPSCs. And here it just shows the different markers that we have looked at both by qPCR or immunofluorescence, just to validate the organoids. This work is actually from two PhD students, two talent PhD student, Fatma Mosmodi, that she's doing the PhD with us, and Nancy Ferreira, that she's at the KU Leuven in Belgium. And they decided to use the gut organoids to study the infection with AV-A71, which is an part of the picornavirus families in enterovirus A71. The reason why we focus on this virus is because to date, there are still no antiviral allowed to to treat this infection. And what Fatma and Nancy did was they developed a toolkit to study the infection with this virus when with the organoids. So they developed an assay to quantify the viral copy number, to assess the efficacy of compounds and their toxicity, and also an image-based readout. So, and here you can see the organoids can be infected with the enterovirus. What was already clear was that in organoids we reach a higher number of copy of the virus already early on compared to a reference cell line. And this goes together with the fact that the organoids are much more sensitive, or the viability is much reduced as compared as when this reference cell line was used. Also, what caught our attention is certain organoids show a very high level of infection, in this case displayed by staining for double strand with an antibody against double strand RNA, while others, they are almost not infected at all. And this gave us the idea to apply a high content image approach to try to understand what's happening in this in the organoids. So basically here we stain the organoids with 3 different markers: DAPI for nuclei, phalloidin for actin, and double-stranded RNA as a proxy of the infection. We took this stack image, and then basically we analyze those images and basically segment the organoids so that we know the numbers, the shape, and the morphology, as well as we were able to count now the number of, in this case, double-stranded RNA per each organoid. And then if you put together all this information, this is some of the data that you can extract. You can actually look at the percentage of infected organoids within one well, within the same experiment, or across different experiments. And what you can see here is, as you increase the amount of virus added to the organoids, as you will assume, the percentage of infected organoids increases. But what really increases is the number of highly infected organoids, here shown in yellow, that are represented with organoids that has more than 15 of these Punkt staining for double-strand RNA. So with all these toolbox that we developed to study the infection in the organoids, we decided to validate this model by applying what we consider a reverse translational approach. So basically, we bring back the compounds that have been tested in clinical trial, and we tested again in vitro using a reference cell line or the organoid model that I've just described. And this is basically to answer the question whether organoids can be a model that can pick up early on drug that during clinical trial has shown either toxicity or lack of efficacy. So in this effort, we tested 3 different antiviral compounds: enviroxime, ropintervir, and 2-C-methylcytidine. They all have 3 different mode of action. They are good in vitro inhibitor, but all of the 3 fail in clinical trial for either toxicity, lack of efficacy, or again toxicity. So in this next slide, if you focus at the bottom, you have the result of both efficacy and toxicity of these 3 compounds. The dotted line show the toxicity. In blue are for the organoids, in red are for the reference cell line, and the continuous line instead shows the efficacy, again, red for a cell, blue for organoids. And what you can appreciate is for enviroxime, there is quite a big shift in terms of potency of this compound when treated in the organoids. And at this point, the activity of the compound is actually start to interfere with its innate toxicity. We didn't see much difference for ropintervir both in cell line as in the in the organoids, while for 2CMC, we start to see quite a bit of toxicity in the organoids compared to the cell line. They were viable at the tested concentration. And in terms of potency, in the cell line, we obtain a nice curve, while when we look at the organoids, we've never been able to reach a full recovery of the infected cells. So we compiled this data all together, and we generate a therapeutic index that is basically a ratio between toxicity of the compound and efficacy of the compound. And the higher this ratio, the better the profile of the compound is. And based on this index, we could show that in cell line, all the three compounds, enviroxime, repintivir, and 2CMC, show a really good performance. On the other hand, in the organoids, both enviroxime and 2CMC show a poor value, while repintivir was still showing a good result. So this is just to highlight that using a more complex system, we could already have eliminated 2 out of the 3 compounds at the early stage of the drug discovery program that instead went through clinical trials. And we can obviously extend this to any other compound where we want to test the toxicity in the gut. So we can look at the proliferation. For instance, here you're looking at organoids that have been treated with staurosporin. We can determine the toxicity. We can look at the profile of the lineage profile of the cell, see if the compounds affect certain specific cell lineage. And then obviously we can look also to whether the intestinal barrier is affected when there is the activity of the compounds that you are testing. So to summarize this part, what I've shown you is that the organoids provide a very attractive model to study the host pathogen interactions in a more complex environment, and also that the organoid provide a more selective barrier towards those compounds that can basically, either because they don't have a good efficacy or they are not safe enough, this can be picked up by using the organoids already early on in a drug discovery program. And this is what we think is the reason why to introduce 3D organoid model at early stage of the drug discovery pipeline. And with this part, I'll conclude, and I will hand over to Dan, who will discuss how to build in vitro model to study the innate and adaptive immunity.
Thank you, Ludo. I'd like to tell you about another way that we can use organoids to improve infectious disease drug discovery, and particularly in the vaccine space. So you will all be familiar, I'm sure, with the COVID-19 vaccines. And the aim of vaccines is to arm an individual with pathogen-specific antibodies or pathogen-specific immune cells. Now, despite the successes that we saw during the pandemic for COVID-19, developing a vaccine can be really challenging. And a point in case is HIV. There's no effective vaccine against the disease, despite many, many years of research. And the reasons are multitude and numerous, but today I'm going to tell you about one of the key ones. So often we won't know if a vaccine works until very late in its development, often after phase 3 clinical trials, when often it's too late to go back and start again. And part of the reason is that actually many of these vaccine candidates would have been tested in preclinical models to see if they work. But those models don't necessarily always tell you how well they're going to work in humans. And part of the reason there is that it's actually very difficult to model human-specific immune responses in preclinical models. So what happens when you receive a vaccine is that the vaccine antigen, be it a viral protein or a bacterial protein, is uptaken by antigen-presenting cells, for instance, dendritic cells, which you can see in this picture on the left-hand side. And those dendritic cells process the antigen and induce either a B-cell response, which will make antibodies against the pathogen, and or a T-cell response, usually through CD8 T cells. And in both cases, you will get memory responses too, where you can potentially prevent reinfection. And part of the reason is that those immune responses happen in something called secondary lymphoid organs. So things like tonsils and lymph nodes. And what you can see from these pictures is these are really complex, highly specialized organs. And if you hone in a bit more into where the B cell responses happen, for instance, within those organs, you can see it's a highly orchestrated and complicated set of processes. So, for instance, B cells mature through successive rounds of expansion and selection. And through a few processes, you end up with hopefully a very mature antibody that can neutralize your pathogen. And some of those features include somatic hypermutation, where the variable genes in antibodies mutate, so you'll generate new antibodies, and those antibodies then be selected for affinity, so the affinity maturation step. And you will also get something called class switching, where you turn into a different version of the antibody, which is the right one. And actually, all of those features are not easily modeled, either in human blood samples that you might use preclinically to test your vaccine, or actually in, for instance, small rodent models, where you will see all of those features, but they are very species specific. So to address that issue, we wanted to build on work that was developed from Mark Davis's lab and Lisa Waggar at Stanford. And they've used a tonsil organoid system. So if you'll remember, I mentioned that tonsils are secondary lymphoid organs. And what they go on to show is that organoids generated from this tissue really faithfully mimic some of the features of the germinal center, which I just told you about. So tonsils are readily available from surgeries. They're often removed, as many of you will know. And to generate these organoids, it's actually a very simple process. So you isolate single cells from these tissues, and then you seed them at high density into transwell culture inserts. And then you stimulate either with your vaccine of choice or an antigen of choice, and then wait just over a week. And what happens is these single cells reaggregate and form organoids. And what's really interesting is that they form structures that are very similar to the germinal center, which you can see in the left-hand side of the slide. And they'll form these light and dark zones, which actually are the parts of the germinal center where you get those successive rounds of maturation. What's also interesting is that those tonsil organoids also comprise many of the key immune subsets that you need to mimic what happens in the germinal center and which are lacking in other models. So for instance, in this particular example, you can see many different subsets of B cells, and they're all really important to include in your cultures. So building on that work, we wanted to know whether we could also see and detect a mature immune response, mature B cell immune response to a standard of care vaccine like the one shown here, and in particular a flu vaccine. And so what we did was a relatively simple experiment where we stimulated these organoids with flu vaccine, waited for around 10 days, and then we looked to detect flu antigen-specific antibodies, so flu IgG, and flu-specific antibody-secreting cells. And what you'll see from the graphs, both for the ASCs and the IgG, is that when you stimulate with a vaccine, all of the donors will respond. But what's interesting is they respond with a different magnitude. And we and others think that that likely reflects what happens in the general population. People respond differently to vaccines. So we think that's a really good thing. Something else that we think is that part of the response we're seeing is a memory B cell response. Most of us have had flu or been vaccinated for flu. And so likely what's happening here is you're activating what's called memory B cells, which are reinvigorated by a flu antigen. However, some of the supporting work that was done from us and others shows that actually you can get a naive response. So brand new antibodies generated in these organoid cultures, not seen before in the donor. And what that means is these organoids support both the somatic hypermutation that I mentioned, and as well as the affinity maturation and the class switching of antibodies. And there's a whole wealth of data to show that's the case. What's interesting is how translatable that might be to the clinic. And really just some of the things you heard in the first bit of the webinar, do they apply here? So if you look at antibody variable genes in the antibodies generated from organoids, what you'll see is the gene usage, so antibody genes you can pick from a number of different genes that come together in the germinal center, what you'll see are the genes that are used mimic what you see in individuals vaccinated with the same vaccine. So the organoids end up generating antibodies that are very similar to what you see in individuals in the clinic. And because of that, we think that we can leverage these type of organoids earlier in the preclinical vaccine development phases as a complementary tool to some of those other in vitro models. Together, they're likely to work much better. And what we hope is that that will better predict how some of these vaccine candidates might behave in humans, and hopefully be part of this iterative cycle of vaccine development that we saw during the pandemic, where we can start to really de-risk some of these candidates and just see how well they work. So I've told you about how you can use these organoids for vaccine development, and it's true that you can actually also use other modalities. So for instance, the mRNA vaccines would work in this system, but we're really interested to develop a way to understand how you can predict immunogenicity of biologic therapies. So anything like gene therapies, cell therapies, or antibody therapies. When patients are dosed with those type of therapies, you will often get an anti-drug response. So anti-drug antibodies, for instance, are generated in those patients, which will neutralize the therapeutic and potentially lead it to not being very efficacious. So we think actually in this system, we'll be able to predict whether that will happen much earlier than you would find out later on in the development phases. And that would be true of any kind of therapeutic that targets infectious disease. So just to sum up some of the key concepts I hope throughout the whole webinar have come across and that we've convinced you of, is that one, clearly organoids much more faithfully mimic the complex nature of tissues, and that makes them ideal for interrogating drug responses in infectious disease, not least because they have the structure, and you could think about drug penetrance here, but also that they'll have all the right cell types to really have that holistic view as to how your therapeutic is working. And I think what we've shown you is that you can probe multiple therapeutic modalities from either vaccines or small molecules in a much more relevant human-centric system, which you can't or haven't been able to do up until recent times. And I think perhaps the key idea to get across today is that these organoids can be leveraged to make better predictions of both drug toxicity, so the gut organoid examples are really pertinent here, and efficacy when it comes to the vaccines. So you can kind of de-risk that drug development earlier on. And I think hopefully what we've convinced you of is there is potential to incorporate organoids into multiple stages of drug development to enhance that translatability and hopefully get those drugs to patients faster. So finally, I'd just like to acknowledge many of the people that are involved in this work from the Charles River side, as well as some of our partners that have helped carry out this work, including Organovir, KU Leuven,